Good afternoon, everybody. We're going to start the next session, which is on shaping metabolism. So for the first speaker, it's my pleasure to introduce Dr. Christos Mantazoros. He is professor at Harvard Medical School. He's the chief endocrinologist, and he's the director of Human Nutrition Unit in Endocrinology, Diabetes, and Metabolism. And today he's going to talk to us about linking energy, sufficiency, and reproduction in mammals, the roles of leptin and activin.
Thank you for the introduction. So in the next 30 minutes, I'm planning to talk to you about the interplay of metabolism and reproduction. I was advised not to talk so much about data, but about a vision, what is coming down the road on the basis of data, of course. So welcome to our world. What we study in our group is energy homeostasis, physiology, and the two extremes, obesity and anorexia nervosa or energy deficiency. And as you know, pathophysiology. And as you know, obesity is related to PCOS or metabolic disturbances and reproductive abnormalities. Anorexia nervosa and hypothalamic amenorrhea, RED-S, is also related to behavior, bone, neuroendocrine abnormalities, infertility. So I'm going to start by talking about leptin, activins, follistatins, and later about the proglucagon-derived peptides. So try to go from epidemiology to pathophysiology to what is coming down the road in terms of challenges, diagnostics, therapeutics. Let me start from energy deficiency, anorexia nervosa, extreme energy deficiency, extreme phenotype of neuroendocrine reproductive abnormalities, very rare, 0.6%, more frequent in females. Hypothalamic amenorrhea, more common in strenuously exercising humans, men and women. And the new term is relative energy deficiency syndrome. Reproductive abnormalities, neuroendocrine abnormalities, immunological abnormalities, and GI hematological abnormalities. I'm going to go over this and link those in a few minutes with hormones. And then the other end of the spectrum is obesity. As a society, we become more and more obese. 1/3 of the population is obese, 1/3 is overweight, distinct neuroendocrine and reproductive abnormalities. The USA is leading the way. We are becoming obese faster than other nations that are following with a lag phase related to the degree of their westernization. So when I was in training 25 years ago, this is 2 paragraphs, was everything that was told about adipose tissue in classical textbooks of endocrinology. The discovery of leptin was the key that opened the black box of energy homeostasis. And today I'm going to talk to you about leptin, GI secreted peptides, and we found that each and every organ communicates with the brain to regulate neuroendocrine homeostasis. So I'm going to talk about muscle also. So when I was about to become independent, my mentor was encumbered with Eli Lilly. So I approached Amgen, which won the battles for leptin patents. And I worked with them. They gave me the molecule, and I got NIH funding to do my studies. So I started with PK studies, pharmacokinetics, to learn how much leptin to give to men and women under what conditions. And this summarizes our findings. So in energy deficiency, leptin levels are low. In obesity, leptin levels are high. And leptin levels are related to neuroendocrine reproductive function, energy homeostasis, and immune function. And when we replaced these in normal people, we fully normalized reproductive hormones and the other neuroendocrine abnormalities to a certain degree. So very fast, physiology in mice is different from that in humans. In humans, leptin is very important in energy deficiency, not in obesity. All the obesity trials failed. And on the basis of our physiology studies, two indications emerged for leptin. One is reproductive abnormalities and metabolic abnormalities in lipodystrophy with low leptin levels. And leptin is approved for lipodystrophy with reproductive abnormalities, extremely high blood sugar and triglyceride levels. And is also important for hypothalamic amenorrhea without metabolic abnormalities. But this, we have completed phase 2 trials in humans, and phase 3 need to be completed. So this is from our epidemiology studies. Anorexia nervosa, hypothalamic amenorrhea, people have low leptin levels. We all know that as we grow older, we go through puberty, we activate hypothalamic peripheral axis, including the reproductive axis. This is disturbed in hypothalamic amenorrhea, anorexia nervosa. The estrogen levels are low, and this leads to infertility, bone loss, immune abnormalities, and other neuroendocrine abnormalities. How do we put all these together? What is the conductor of the orchestra that gives the brain the signal there is no energy in the periphery? This is leptin. When leptin is missing, the conductor is not there, the orchestra is disorganized, and we see all these abnormalities. We proved this in at least 2 papers. One was the original paper, uncontrolled open label in the New England Journal of Medicine, and then we had follow-up papers in PNAS a few years later. What did we prove? That by giving leptin, we fully normalize reproductive function, and then through either estrogens or other hormones and indirect mechanisms, we improve bone metabolism. And if we study those women long enough, 9 months or 2 years, where our previous studies, randomized control studies for 9 months, we improve bone mineral density and bone mineral content by 6.5% in 9 months. So what remains to be seen is what leptin does in the brain of humans. You will see a lot of data from mice in the hypothalamus. The hypothalamus is the homeostatic system. This is important, but most of us don't become obese because the homeostasis is wrong. I mean, the reward system in humans, the cortex, making decisions is very important. So this is one of the focuses for the future. In humans, we have to do studies beyond what we learn from mice in terms of the brain and energy homeostasis to get the picture in humans. What else remains to be seen? What remains to be studied is immune abnormalities improve in response to leptin, but hematological abnormalities are not fully explained by leptin. And then there is also a question about activin follistatin that we are actively exploring in the lab, and we have good findings. Now, what all of you know the role of activin follistatin inhibin system in the reproductive system, right? Activins act at the level of the hypophysis, and they also act in the periphery. But we have found that activin and follistatin are also important in terms of muscle growth. So activin myostatin block muscle growth, and follistatin blocks activin and myostatin. And this is an animal model of mutations in the myostatin gene. There is similar pictures in humans with very muscular humans. So very briefly, we studied over the past few years the physiology of activins and follistatins in humans. We are finding that, again, similar to mice and animals, activins regulate muscle growth, increasing follistatin regulates muscle growth. And then what happens is that in not only the reproductive system, but activins and follistatins regulate muscle and regulate glucose homeostasis and insulin resistance. How do we put all these together? So we wanted to see what happens in energy deficiency, right? Hypothalamic amenorrhea, anorexia nervosa, energy deficiency. We know that low leptin blocks the hypothalamic pituitary peripheral gonadal axis. What is the role of activins follistatins? Is it independent from leptin, or is it through leptin? In brief, series of papers that we published in the past few years on acute energy deprivation, we found that as leptin goes down, GnRH, LH-FSH goes down, estradiol goes down, we have hypothalamic amenorrhea. Again, activins go down, follistatin goes up. And this blocks the system, so blocks the reproductive system. Makes sense not to procreate when we don't have energy. But this axis, this pathway, is independent from the pathway of leptin. So we have two pathways. One is coming from fat, low energy in fat, gives the signal to the brain to shut off the reproductive axis. The other one is the activin follistatin system. A similar finding in chronic energy deprivation, hypothalamic amenorrhea and anorexia nervosa over months. The same systems, like both systems inactivate the reproductive system. And what does the activin follistatin system do at the same time? It tries to preserve muscle by creating relative muscle growth and causes insulin resistance. And through insulin resistance, it causes hyperglycemia and lipolysis. So energy is coming, every drop of energy in fat is coming to the bloodstream to be used for the survival of the organism. So these are the two main axes, in our opinion, that need to be leveraged as therapeutics in the future, right? So we need to find, are there any other hormonal axes, especially in terms of hematology in anorexia nervosa or red S syndrome? Are there other mechanisms? Number two, can we go into phase 3 trials with leptin administration, adjusting the doses so that we don't see weight loss, right? So there is a hierarchy of responses. As we grow from zero leptin to go up, we normalize immune function, reproductive function, and if we keep levels to the normal range, we don't see any weight loss. How can we leverage this as treatments in the future? Let me move now very briefly over to the other end of the spectrum, obesity. The prevalence of obesity goes up. Over time, the prevalence of diabetes goes up 15%. The prevalence of PCOS, NAFLD, goes up. So reproductive cancers that are related to obesity. What are the mediators? We think that adiponectin, low levels of adiponectin, are very important in this constellation of metabolic abnormalities that we call metabolic syndrome. And this does not happen in each and every obese person. 25% of the obese people have no metabolic abnormalities. They have high leptin, which they are tolerant to, but no normal adiponectin, normal insulin sensitizer. 25%. 75%, there is not enough depot storage space in the sub-Q adipocytes. So the fat that we are not going to use in the near future has to go somewhere else and spills over to muscle and causes insulin resistance, to the liver, causes fatty liver disease, and this creates a vicious cycle of insulin resistance that leads to cardiovascular disease, chronic kidney disease, and polycystic ovarian disease, which is a part of metabolic abnormalities. We are learning more and more about post-receptor insulin pathways that are related to PCOS. We are learning more and more about metabolomics, lipidomics, and gene networks that regulate PCOS. The question for the future, in my mind, is are we going to find a solution for PCOS through studying these complex genetic, genomic, proteomic pathways, or is it as simple as treating obesity, right? So is it the complex pathways independent from obesity, or if we treat obesity with the new medications that we plan to utilize and reduce fat mass, are we going to treat PCOS? And my final few minutes, I'm going to talk about obesity per se, right? So this is the villain in the village. right? So the underpinning, we understand there is significant, it's not a lack of will, significant hormonal and metabolic pathways. There is an interaction between environmental, societal, hormonal pathways that we need to understand better. Continuous increasing trends, but we start understanding what is happening. And we start developing tools to treat obesity, and through obesity, treat infertility, hopefully, and PCOS remains to be seen, right? So again, this mouse model of leptin, leptin opened the black box. Obesity is positively related to leptin, so there is leptin tolerance. Leptin does not work in obesity, right? And we have shown this many, many times, and this is from one of our review papers, that it does not work in syndromes associated with obesity. However, other molecules that we are studying, which are secreted not from fat, but from the GI tract or the muscle that I told you about, are also useful for the treatment or for understanding and the treatment of obesity. And this is a molecule that we are studying a lot in the past 2 or 3 years. It's preproglucagon. This is secreted from either the pancreas or the brain and the GI tract. Different molecules cleave this preproglucagon to proglucagon and other peptides that circulate in the bloodstream or act locally in the brain, and they regulate glucose homeostasis and metabolism through the pancreas, but also through the brain, they regulate appetite. So oxyntomodulin glycendin is the future, GLP-1, GLP-2 is the present, and the glucagon. So we are developing now medications that are GLP-1 analogs that act in the brain to decrease food intake and decrease obesity. Dual agonists with glucagon is coming down the road, and oxyntomodulin glycendin are still in preclinical stage. So this is from our studies with bariatric patients. We have studied physiology extensively in normal people, obese, lean. This is from bariatric patients. What are we finding is that these molecules, after bariatric surgery, which is very effective in resolving not only body weight, decreasing body weight, but resolving diabetes. 80% of diabetics don't have diabetes after bariatric surgery. And all the other NAFLD, hypertension, PCOS, you name it. These molecules, these GI secreted molecules, go up after bariatric surgery, and we have now 10-year long data. We go 1 year, 5 years, 10 years after bariatric surgery, the levels remain high, and the area under the curve, the higher the levels of these molecules, the lower the chances that someone will relapse in terms of body weight gain or metabolic abnormalities. We have identified the receptors for GLP-1 in the brain. We were the first to identify them in several areas of human brains. And then we did functional MRI studies with lean and obese and diabetic subjects, showing to subjects either highly desirable foods or less desirable foods or non-food cues, trying to see before and after we give these hormones, trying to see what is activated and what is not activated, right, in humans. We see some activation in the hypothalamus with leptin and these GI secreted molecules, but most of the areas that get activated are the reward systems. So this indicates to us, and to a lesser degree, the prefrontal cortex. This indicates to us that these molecules act primarily by making food more rewarding earlier. So when we give these medications, people feel more rewarded, they stop eating. And of course, the prefrontal cortex makes us like more vigilant in terms of what we eat. For example, during a break, we go out, there's plenty of food. Some of us are more careful, more vigilant, right? Others graze, right? And they take more calories. So these medications make us more vigilant. All right, are there analogs in development? Yes. Modified GLP-1 analogs like liraglutide are available in the clinic. Semaglutide is coming to the clinic, and semaglutide, which is the strongest GLP-1 analog today, along with behavioral modification, leads up to 15% weight loss. With dual or triple that we are developing as we speak, we think that body weight will go up to 20, 25%. The goal is to reach 40% that we reach with bariatric surgery. So it remains to be seen what the effects of this 20, 25% weight loss is going to be on the reproductive system, PCOS, infertility. So these medications are coming. They are in development, under investigation for diabetes or NASH, obesity, but also for PCOS. And they come in different forms, injectables, oral preparations, sub-Q, osmotic mini pumps. We'll see what the future is, but I think the future is going to be either injection once a week or gene therapy that will last longer, or subcutaneous osmotic mini pumps if we are able to fine-tune it, that this treatment will last for 6 months or 1 year. All right, to summarize, so all of us have a set point in terms of our body weight. Our set point, unfortunately, goes up as we grow older. Our energy expenditure goes down. But for a certain period of time, right, of weeks or months, we keep our set point. We may overeat during Thanksgiving, right, which is coming in a few days, right? But a few days later, our body will bring us back to our original body weight. So what are the main regulators that I spoke about? As we lose weight, leptin goes down and tends to bring us back by activating neuroendocrine mechanisms. As we gain weight, if the system works well, incretins, GI secreted molecules like GLP-1 and others will bring us down, right, to the original body weight. SGLT2s, I didn't have time to speak a lot, also have the same effect. And we leverage now this newly found knowledge of physiology to create novel medications. So similar to leptin that I told you about, SGLT2s in the kidneys, GI secreted hormones, and we have medications now in development to leverage the activin follistatin axis to grow muscle, decrease body weight, improve metabolism, and improve reproductive function. So a lot is going to come in terms of pharmacotherapy in the next 10 years or 20 years. And I think that we will see spectacular responses in terms of body weight and in terms of reproductive function. And I will close with this slide. If our target is not diabetes or NAFLD, but reproductive function, we need to study better and understand better the interplay of CNS, the GI system, fat, white, and brown adipose tissue, as well as skeletal muscle and kidneys, and see how these interact in humans to regulate not only energy homeostasis, but also reproductive function. And we will be leveraging this knowledge now with medications, pills, and injectables once a week to decrease body weight, improve reproductive function in both ends of the spectrum, anorexia nervosa, hypothalamic amenorrhea, and obesity. Also, nanotherapy and gene therapy are being studied. All right, so some of the questions, and I will close with this slide. Obesity and its comorbidity, precise targets in genetically defined obesity. We are developing medications that attack these genes, very, very small percentage in a great percentage of patients, the large percentage of patients who don't have genetic mutations, behavioral modification, new medications that leverage the GI and muscle secreted hormones. It remains to be seen what is going to happen in terms of PCOS reproductive abnormalities. In terms of hypothalamic amenorrhea, anorexia nervosa, we need to see leptin advancing to phase 3 clinical trials for Rett syndrome and hypothalamic amenorrhea. We need to see medications leveraging the active follistatin system. We need to understand better what happens in human brains in terms of the wiring, the pathways in the brain, hypothalamus and beyond. And we need to study PCOS in terms of epigenetics, metabolomics, but ideally, we would like to see PCOS being treated with the new medications that we have for obesity. And the role of NIH, industry foundations, or donors is of major importance here. In my opinion, NIH is funding less human studies than they should be funding. So in terms of policy, I would recommend the NIH funds more human studies. The biology is not exactly the same with that in mice. And I'm sorry for my colleagues who study mice, but mice are nice, flies are nice, but humans are not mice. We have had a balance between NIH and foundations and funding from donors. And I found in the past that for my group, we made more of our discoveries with money from foundations and donors without downplaying the role of NIH. So anyway, we need more studies in humans, and we understand the pathways, and I think that we are going to have exciting discoveries and exciting developments in the next 10 to 20 years. And this is what I was asked to present, and I hope I gave you an overview of what our focus is and what I think is coming down the road. And thank you very much for your attention. I'm exactly at 30 minutes. Thank you.